Hi, everybody. Welcome to Love Goes Live. My name is Mark Lupquist. I'm a molecular biology professor at St. Michael's College, and I'm making videos for my students in the St. Michael's community and really for anybody who loves science on COVID-19 and other sciencey stuff. And a good friend of mine in Portland, Oregon asked me to look into the molecular biology on the origins of COVID-19. So without any further ado, let's get into it. So let me share my screen with you as I always do. And I just learned how to use a new fancy feature. You'd think I would have known how to do this for years, but the laser pointer. Okay, here we go, my laser. So when I started to look into the literature, I found a paper that was published in Nature. And if you remember, Nature is one of the most premier journals in the science field. This was published back in early February, which means that it was submitted for publication probably in mid-January. So this is one of the first papers that postulated what is the origin of COVID-19, where did it come from? And if you've seen the other Love Goes Lives, what they did was what's called a phylogenetic analysis. And what they do is they sequence all 28,000 nucleotides of the COVID-19 genome. And they did that for multiple clinical isolates. So these are viral samples that they took out of people in Wuhan, and then they sequenced the RNA genome. And then they asked just a very simple question: what's it most similar to, or what's it most identical to? And these trees, when you look at them, what you want to look for is distance or branch length. So anything that's really closely related will be right next to each other. So these all have the same branch length, all these isolates, and they should because they are the same strain. And then the closest relative is this coronavirus that's from a bat. And then if you look at the second closest and third closest relatives, notice they're also coronaviruses from bats. In fact, if you just scan through this whole tree here, there's a lot of bats, and it's not until you get down here that you start to see some human coronaviruses. So the bat that it was most similar to, or the coronavirus, I should say, from which the coronavirus to which is most similar came from this flying furry creature over here, which is the horseshoe bat. All right. And you look at identity. So there's 28,000 nucleotides, a little north of that, in the COVID-19 genome. And when you look at identity, it's about 96% identical to the coronavirus that is found in the horseshoe bat. And that's pretty good. So that's pretty convincing evidence that the virus came from the bat. But another group asked a very simple question. Coronaviruses don't mutate very quickly. So what about the 4%? So if it jumped from bats to humans, it should be like 99.9% identical. So something's up there. Like, where did that extra 4% go? Because it wouldn't mutate that quickly. And as I started digging around, I need to go over just a little bit of biology from some previous Love Goes Live, so this will make sense. So this is an image from the journal Science. It's on the cover. This beautiful, beautiful picture right here is ACE2. So this is the receptor, the viral entry point in human cells. This is the virus right here, COVID-19. And coincidentally, if you're curious, the reason it's called corona is in crown, because the spike protein, which is right here, my little highlighter, is thought to look like a crown. So the way this works is the spike protein fits into this little notch here of ACE2. So it's a structure-function relationship, and the pieces have to fit. All right. So this group actually looked at the human ACE2 receptor. This is what it looks like in the real world, as I would say. And I'm sure this makes it so much more clear, right? And then this is the viral spike protein. And really they're asking the question, when we zoom in and look at the interaction, do the pieces fit? And the answer is, when you look at the bat coronavirus, the one that is most similar to COVID-19, the pieces do not fit. In other words, the spike protein from the bat coronavirus does not fit into the pocket of ACE2, which means it shouldn't be able to get into human cells very easily. All right, so that got this group thinking, and they actually wrote a paper which they published in Cell, which I have referenced here. Cell is also one of the premier science journals, so pretty high bar to get published in Cell. And they did a similar analysis. So they took, here's the clinical samples. Once again, they sequenced 28,000 nucleotides of RNA. They found the closest relative is that bat genome, that bat coronavirus, which was the horseshoe bat. And then when you put pangolin coronaviruses in here, it's the second most closely related coronavirus. And if you're like me, you're probably wondering, what is a pangolin? So a pangolin is this actually quite adorable creature that's over here on the left of your screen. And it actually happens to harbor coronaviruses. And so what this group thinks is that COVID-19 came from a bat, At some point, it passed through a pangolin, and the pangolin, coronavirus in pangolin, recombined to create a third virus, which we now call COVID-19. So let me say that again. So the group thinks that it came from a bat. 96% of it seems to be identical to bat sequence, but then a small part of it seems to have come from part of a coronavirus that comes from this very adorable creature that they call pangolin. Now, the real question is, where did that 4% come from? Now remember, the bat coronavirus, its spike protein doesn't fit into the ACE2 receptor. So the working model is, and if you look at the part that seems to have come from pangolin, it's what encodes the spike receptor, the spike protein, sorry, not receptor, the spike protein. So the spike protein seems to have come from a pangolin, and the rest of it seems to have come from a bat. All right, let me stop sharing here. So what does all this mean? Well, this means that I think it pretty clearly demonstrates that the COVID-19 originated in a bat. At some point in this history, it passed through a pangolin, or maybe it just recombined with the pangolin coronavirus. And now that a little piece of that pangolin RNA genome ended up in COVID-19, which enables it to infect humans via ACE2. Now, I know what you're wondering, because everybody asked me this. Was it made in a lab? And the answer is no, it was not made in a lab. There doesn't seem to be any evidence of that. Every paper that I read referenced that it was not made in a lab, and there are certain signatures you would look for if it was made in the lab. You'd look for viral backbone, the type that has been published before. You'd look for certain editing features for when you do recombination, or where you actually go in and do genome editing. You leave little fingerprints behind, and none of those have been found in COVID-19, which suggests or demonstrates that it was not created in a lab. So take-home message, bat, somehow mixed with a pangolin, made a chimeric RNA genome that we now call COVID-19. And how it got into the first human is still a little bit of a mystery. Okay, that's all I got for you today. My name is Mark Lupkowitz, trying to rise from the ashes like the phoenix and let crisis bring out the best of me. All right, have a great day. And next up on Love Goes Live, pets and COVID-19. What does the science show?